Grade 12 Mindsets, welcome to it. This is Learn Extra Live. It is spring school, so we are learning more still. You've been waiting for this lesson, so we bring it to you. I am Abram, and I'm not alone. I'm with Aslam. It is A squared day. How are you, Aslam?
A squared? Huh?
Yes.
From AB to A squared?
Yes.
I'm great. I'm Graham Abram.
You look
Welcome,
different, by the
Mindsetters.
way.
Different?
Yes.
I'm still looking with my eyes.
Change is good.
Change is good, huh? Good.
What are we doing
What
today?
are we doing today? Today we're talking about paper one stuff. But mainly, you've heard earlier, meiosis, DNA, and we move on in the chapters to genetics and biotechnology. Please understand that biotechnology is not separate from whatever else you're doing, but more about it as we go along.
Looking forward. I have my genes on to learn more on genetics. Otherwise, Mindsetters, if you'd like to learn more with us, it's simple. Join us on Facebook, like our page. It is facebook.com/LearnExtra. Join about 53,000 people that are on the page and be amongst them. Otherwise, you can follow us on Twitter @LearnExtra. And lastly, download your notes all for free on learnextra.co.za/spring school, all in one word. Otherwise, the first notes link will be posted on our Facebook page. And we have an exciting competition that I need to tell you about right now. We have a clock that has started right now. All you need to do is to tag yourself on the post that is posted on Facebook on the number that you like. For instance, what is your favorite number from between 0 and 9, Aslam?
3.
It's 3. Aslam's number is 3. So if your number is 3, you're going to take yourself on 3 on that board. Otherwise, share that post on your own favorite, on your own wall, and then write 3 on that. Comment 3. My lucky number is 8. So I'll go for 8. So that is an example. So I'll be telling you more on the competition as time goes by. You could win a mystery nice package. Over to you, Aslam.
Thank you, Abram. Welcome, Mindsetters. We are on the page. We are moving. You know there are not many days left for your life science exam, national exam. Now that's everybody across the 9 provinces. Everybody writes on the same days, the 15th and the 18th of November. So that gives you less than 2 months to go. It's time to start getting the pace a little bit faster now. You started with your revision. If you haven't started, this is the time to start. Get moving with these little programs and the past papers. So we're looking at genetics and biotechnology. We just want to again tell you here what we need to do very clearly. Here would be, number 1, you need to know a little bit about Mendel and his work. And with that, you need to do a little recapping on terminology. The terms used in genetics, and all you need to do is go to our live earlier in the year when we did genetics proper as a topic and see that we started with terminology and we stressed those terms. So you need to go through those terms over and over again to get an understanding. Without the terms in your mind, guys, you may not be able to solve the problems that come thereafter. So learn the terms properly. Then we move on to the types of problems. And in the problems, we look at straightforward monohybrid. You only have to do monohybrid. Under that, complete dominance, incomplete dominance, and codominance. So you need to learn how to solve problems in these 3 types of dominance. You also have to look at, further than that, you need to look at sex linkage. What is sex linkage and how do you solve problems that involve some form of sex linkage? This is what you need to do at least in these things. You also have to look at blood groups and the inheritance thereof. In a nutshell, guys, these are the important parts. Doesn't mean you have to leave the other things out, but these are the things that are tested more often. And these are the things that children have problems with, especially this one, and this one here as well. the blood groups and the sex linkage. We have problems on that today. We're going to try and solve those problems. At the same time, I want to show you today that solving these problems, this one and this one, is still based on the same framework as one would solve a problem on complete dominance. In fact, all of them are solved on the same template, on the same recipe, on the same method. One just needs to get the hang of it and then move on from there. Good. Let's not waste time talking about that. Let's move on with the questions. And the first question then coming from the Eastern Cape Prelim 2013. That means the trial exam written in the Eastern Cape this year. We are using fresh questions here. The diagram below shows a genetic cross between rabbits. Now, watch what I'm doing with the pen as I go along. The white fur color is dominant over the dark gray fur color. So they're already telling you which one is dominant over the other one. Okay? And at the same time, it tells you if one is dominant over the other, that means this is an example of complete dominance. One factor is completely dominating the other one. Study the diagram and answer the questions that follow. Use the symbols. They're telling you using the symbol, use the symbols capital W for dominant, uppercase W, and lowercase for recessive. Generally, people, it's not a very good idea to use these two letters in solving complete dominance, simply because, especially when you're handwriting it, it becomes a problem sometimes to distinguish between the uppercase and the lowercase. If you have a problem, however, with W used in the question, then you make sure that you try to make the marker understand which is your capital W. That means your big W or your uppercase W must be much bigger than the lowercase w, so the person can distinguish between the two. Remember, it's not like other letters like B, capital B, small b are different, R and R are different, and so on. But W and W, same story there. Good. Just some background on that one. So they're telling you, here's a female parent and the male parent. Here's white, and the male parent is gray. Now, look at the offspring. Always, when they give you a diagram like this, or a pedigree diagram, the way you work it out is absolutely easy. Let me show you what I mean by that. Here's a female parent, here's a male parent. This one is white, as you can see, and this one is gray, as you can see. Now, look at the offspring coming from these two parents. We have some white ones and some gray ones. What does the question tell you? It tells you that white fur color is dominant. So in other words, this rabbit here, this female, must have had a capital W. But always easier, but we don't know what's the other letter here. I'll come back to that just now. What is the easy thing to do? The easy thing to do is to identify the recessive one. Why? Because the recessive one can only be homozygous recessive. Remember the story, when the cat is away, the mice will play, the law of dominance. That means if the dominant gene is there, then the recessive gene will not show. What does Mendel say about this? He says when two characteristics are put together and the one is dominant, then the dominant one will mask or shadow the recessive one. In other words, the other one will not show. Hence my analogy, the cat is there, the mice won't play. When the cat is away, the mice will play. So using that analogy, the gray is easy to do then, because the gray has to be homozygous for the condition. So the minute the one gray is like that, all the grays are going to be that way. Notice I'm using the curve to show the difference between the uppercase W and the lowercase w. And at the same time, we know for sure that these ones must have the uppercase W. But now the question asking us is, will these animals be homozygous? In other words, will they be homozygous for white, or will they be heterozygous for white? This is the question you have to ask. And notice the homework we're doing before we even looking at the question. We are simply looking at the diagram. And once we do this little homework, spend 5 or 6, 7 minutes doing this, the rest becomes easy. Now let me explain why. Now if this guy has 2 small w's, that means this male parent would have given all of his offspring one of 2 options, and his 2 options are only small w, small w. And hence we see that there. Now look at these children. They've got this one small w from the father. Where did they get the other small w from? Obviously from the mother. So the mother had to have a small w as well. So that's where that other w comes from, the small w comes from. And therefore, and the fact that these are white, they must have the capital. But because their father was homozygous for gray, they would have inherited that from their father. So therefore, they are also heterozygous. That is how we can see whether an individual, especially the dominant one, is heterozygous or homozygous. So first, you go for the recessive. Even in a pedigree diagram, go for the recessive first. Now let's move on. Represent a genetic cross to determine the possible genotypes and phenotypes of a cross between a white male rabbit in the F1 generation and the white female parent with a genotype which is the same as that of the white female represented in the P1. So that is why all that homework was necessary. There's the genotype of the female there, and the white male, there's the genotype given there. Okay, good? Now let's see how we solve the problem. And I want you to pay special attention to the layout, because the layout tells us about how you earn your marks here. And there we go. We start by writing down P1 and write the phenotype on the top, because when you write the genotype, then your problem will follow from there. Phenotype, they are both white. Notice, you must have all that correct to get that 1 mark. Then genotype, both are heterozygous. And you must have again all of that to get that 1 mark. Thereafter, you'll put the word meiosis. You can put it there. I prefer to put it there. And what does that mean? That meiosis takes place to split the genes into different gametes. That's what we are showing here. Notice that you have to separate those genes. Or rather, separate the alleles. Remember, this is one gene separated into 2 alleles. And the same on the other side. You must show them apart, because that's telling the examiner you understand that when meiosis takes place, you split it. Notice how your meiosis knowledge now is important. Thereafter, you write the word fertilization. And you can put that here as well. If these 2 words are written in the correct place, then you would get a mark for that. As you can see, the examiner has shown it there. And if P1 and F1, after that, in line with your results, you'll show the F1 generation. Remember, you don't have to use the lines. You could have used that as well, the Punnett square, which is neater and easier for yourselves as well. So what do we do in the Punnett square? We separate those 2 genes, alleles again, and our offspring, uppercase W, uppercase lowercase w, uppercase lowercase, and lowercase lowercase. So what have we got here? We have got phenotype, or here's the genotype, homozygous dominant, heterozygous, heterozygous, homozygous. 1 is to 2 is to 1. And at the same time, we have 1, 2, 3 white is to 1 dark gray. That is our phenotype ratio. And generally, people, whenever we do a cross with 2 heterozygous individuals, we will always get this result in a complete dominance question. Whenever we do a cross between 2 heterozygous individuals, we will always get 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive, and 3 of the dominant color and 1 of the recessive color, or 25%, 75%. If the question asks you for percentages, you talk about percentages. If the question asks you for a ratio, then you give ratio. You notice that we spent a lot of time dealing with the way we set out the problem and showing you how the marks are allocated. You need to keep this in mind. Remember, if you, for some reason, this is normally about 6 marks. So if you had your format correct, and this part would be easy because you would have got 1 mark for the phenotype is easy, it's given in a question. So you'd have got 1 mark there, you would have got 1 for P1, F1, and you've got 1 for meiosis and fertilization. Even if your letters all here are wrong, you would have got 3 out of 6, which is 50% better than nothing. So make sure that you follow this format with all the problems, and we're going to emphasize this as we go along. With that, a B.
Thank you so much. I just want to say this on the competition, guys. We're currently on 53,784. So the last digit is 4. If you like Aslam, you've chosen 3. It means you need to get 9 of your friends to like the Facebook page. And if you're like me, you've chosen your 8. You just need 5. You need 4 more friends to like the page. Otherwise, after the break, we'll be telling you more on the competition and learn more. Learn extra. Don't move. Welcome back, Mindsetters. Now, we'd like to hear from you. How are you enjoying the show? Your questions, right on Facebook. Like the page. It's facebook.com/learnextra. But now let's take a look at some of the questions that came through. Aslam, are you ready?
Ready as always.
As always, right. Let's start with this one from Reflector. I always find questions that say you must represent a genetic cross. So I don't know if I'm allowed to use the Punnett square to show the phenotypes and genotypes.
Reflector.
Yes.
Yeah, let's reflect on that, Reflector. Okay, Reflector. The options are given to you. Notice here, this particular examiner favored this one here. Okay, so he's showing the line. Most of them show this on this side because it's easier to just stick this one on the other side. Remember, even if you don't do all of this and you just put this below, you take the whole Punnett square and you put it below there, you're getting your full mark. So it doesn't really make a difference whether you're using a line, genetic diagram, or a Punnett square. Examiners can't tell you to do one or the other. They will tell you draw a genetic diagram. Next question.
Maureen says, may you please repeat that genetic cross?
Maureen, we don't have much time to repeat this genetic cross, but we've got so many after this that you should get into it. And if you still don't get it, at the end, towards the end, ask again, maybe we'll have time.
All right, last one from now. Shikash, may you please explain the Mendel's 2 law?
Shikash wants to know Mendel's 2 laws. The 2 laws that we concentrate on are law of dominance and segregation. There's one more, the law of independent assortment. It's not so important for you because you don't do dihybrid crossing, so I'm not even going to go there. Most cases, when we explain that one, many learners confuse that with a random assortment. Independent assortment and random assortment are 2 different things. We'll talk about that just now. But more important, the law of dominance states clearly that when 2 characteristics, remember Mendel did not know anything about genes or meiosis. So that's why we're using his law. When 2 characteristics are together and one is dominant, the dominant one masks or shadows the recessive one. In other words, when they're together, the recessive one will not show. That's what it means. First understand it, and then try and word it around that. The important thing is when 2 characteristics are together, and one is strong, dominant, and the other one is weak, recessive, the recessive one won't show. That's what it means. And the law of segregation, here Mendel said for each characteristic, there are 2 factors. Every characteristic is controlled by 2 factors. What does it mean segregation? These factors separate in sex cells. Remember again, Mendel did not know about meiosis. So what can we say in our time today? We can say for each characteristic, there are two alleles. These alleles separate into different gametes during meiosis. We can say that in our case here. Good, we're moving on. If there's other questions, we'll take them as we go along. Question 2 now. Now we're going to go faster with some of the questions now because we have got rid of the format issue and the explanation that goes with it, okay? Now a cross between a white cat and a black cat produces a white cat with black patches. So here we have two different characteristics, black and white, and the individuals that form from this union have both characteristics in them. Already that means that neither black nor white is dominant over each other in this particular example. That's the first thing. Now when there's neither of them dominant, that means we rule out complete dominance out of the equation. Now what can it be? It can be incomplete dominance or codominance. Now how do we know what's the difference? When the individuals show both the characteristics, black and white, this is codominance. Why? They're sharing it. When the individuals show another blend of a color, for example, black and white, and we got gray out of it, or red and white, and we get pink, or brown and yellow, and get orange, whatever it is, but a new color, a new characteristic, then it's incomplete dominance. So this is what you have to look at. When neither are dominant, that means it can't be complete dominance. It can either be incomplete or co. If both show in the individual's offspring, then it's codominance. If a new blend of color or characteristic comes there, you know, tall and short, medium, for example. It's a ridiculous example, but just to give you the idea around it, that's what we are saying. Then it's incomplete dominance. So let's see now. So what type of here, what's happening here? There's both there. So what type we said? In this case, it would be codominance. Why? Obviously, the next question is going to say, give a reason for your answer. And the answer is, none of the characteristics are dominant. That's the first thing, because whether it's incomplete or co, that will still be there, that none of the characteristics is dominant. That's the first answer. And the second mark, both phenotypes are visible in the offspring. Both phenotypes are visible in the offspring. Represent a genetic cross to determine the possible genotypes and phenotypes of the above cross. You'll notice I've repeated this in all the questions because we want to pump that idea to you. How do we get to that answer? And here now look how we have set it up again. Same story again. We start with our story of P1, phenotype, black and white. This is what happened. Now notice the genotype. Because neither was dominant, we used two uppercase and we used the individual's colors. That means B for black, W for white. You can use whatever you want to if the question does not prescribe it, but you must provide a key to tell the examiner. But if it's co or incomplete, you cannot use a capital and a small. You must use capital letters to show that they are equally dominant. That's important. In complete dominance, you must use a capital and a small to show that one is dominant and the one is recessive. Again, your meiosis is given there. Your gametes separated again. Your fertilization. Notice I've only done one because they're all the same. Whether we've taken each one here, we'll get all capital B, capital W. And here's the one with the Punnett squares. You could have done that. Capital B, capital B, capital W, and there's your answer. Capital B, capital W, all heterozygous. So your genotype is 100% heterozygous. And phenotype 100% black and white. Can you see the way you work out the problem, whether you're working incomplete, complete, co, the way you work out the problem is the same, is only the way the letters are used are different. Good, I'm hoping you get that one. By the way, even if this was incomplete, if this was incomplete, for example, we had black and white, and our answer was that the offspring were gray. The way you worked out the problem up to this point would all be the same. Only your phenotype answer right at the end would be 100% gray. That would be incomplete dominance. So you'll still keep the capital B, the capital W. All right? So in this example, we have done both codominance and incomplete dominance, if it had to come that way. Why am I going backwards? Good. We're moving on to question 3. We're moving now. That one took a bit less time than the first one. Okay, in rabbits, assume that the dominant allele B produces black fur, and the allele small b or lowercase b for white fur is recessive to capital B. So they're telling you here that black is dominant and white is recessive, and therefore we use capital B and small b. What type of dominance? We have to ask ourselves. Complete dominance, because there's one that's dominant and the other one that is recessive. So what color fur will each of the following rabbits have? This is a very basic, but guys, bear with me. If you can understand this particular question, then no genetic problem can phase you after this. They want to know, rabbit 1, the genotype is capital B, capital B. So obviously that one will be black, based on what they told us earlier. Capital B, small b, phenotype would be black still. Whether the small b is before the big B or the big B is before the small b, your answer is still black because it has both. All right? And the last one, a homozygous, oops, undo. The homozygous small b's, lowercase, will give you white. This we have discussed in our earlier example as well. Which of them will breed true? Or which of them are true breeding? Or which of them are homozygous? This is what this question wants to know. From these here, which ones are homozygous? Which one are true breeding? That's what we are asking. And they are obviously the 2 capital B's and the 2 small b's. The next one says, which rabbits are homozygous for coat color? Which rabbits now? Rabbit 1 and rabbit 4. Notice they're trying to, again, what is this question trying to do? It's trying to establish whether you understand that homozygous and true breeding is the same. Now the opposite to that would be heterozygous or hybrid. Understand that? So it's 1 and 4. Rabbit 1 and rabbit 4. If rabbits 1 and 4 were mated together and had 12 babies, how many of these would you expect to be black? Rabbit 1 and rabbit 4. 2 capital B's homozygous with 2 small b's. We've looked at that example earlier. Whenever we get homozygous dominant with homozygous recessive, then all the offspring would be the dominant color. And therefore, our answer would be 12 out of 12. If rabbits 2 and 3 are interbred and produce several litters totaling 48 babies, how many white babies would be predicted by the laws of genetics? So if they had 48 babies, and they want to know how many white. Remember, white is recessive, so 2 and 3. 2 and 3 are 2 heterozygous. What did I tell you earlier? Whenever we have 2 heterozygous individuals and it's complete dominance, then 25%, a quarter of those would be white, or the recessive color in that case. And therefore, the answer would be 12 again here. Remember, don't relate the 12 to the 12 above. That 12 was for all, 100%. This 12 is a quarter, or 25% of 48. If rabbits 3 and 4 are mated together on several occasions and have 50 babies, how many of their babies would you expect to be black? So what's this again? 3 and 4. 3 and 4 now, you have to look at that one there. It's heterozygous and homozygous. It's not a straightforward one here. Now, you have to obviously work out a Punnett square for that, and it's easy to do that. Simply draw a quick Punnett square on your question paper. Heterozygous was a 1, 3, and homozygous recessive was 4. Capital B, small b, capital B, small b, all right, that's giving me a problem there. All right, there we go. Capital B, small b, and these ones, capital B. Am I right? Abraham, are you checking my work? So, if 50 let's go back. I just want to check one thing quickly. That's number 3, yes? And that's number 4. Have I put the right thing there? There we go. That's number 3 and that's number 4. Oops. Things are moving, Abraham. How many of the babies would you expect to be black? Black is dominant. How many? 1, 2. But did they ask you out of 4? No, they asked you out of 50. So, but how many percent is that? That is 50%. 50% of 50 equals 25. So your answer would be 25 would be black. I'm hoping we get the answer. Notice this now, a totally different spin on genetics. So far we've asked you to work out a genetic problem. Here we're giving you information and we're asking you questions. Keep that in mind. A, B.
Thank you so much, Aslam. And about the competition mindsets, before we take the break, remember you can either tag yourself on that post or share it on your wall and then write your favorite number. Like for instance, I chose 8 earlier on and Aslam chose 3, and currently we have 53,798. So if we were to stop the clock now, I'll definitely go through to the draw because the last number or the last digit is 8. So mindsets, that's how we roll. The prize will be revealed, or rather the winner will be announced on the last show, which will be from 4 o'clock. Otherwise, keep it tuned in. We'll see you. Welcome back, Mindsetters. Now, let me check to you on the page and just see what are you guys saying. Aslam, we have Maureen saying there's a mistake on the genotype of paper 1 on that example of CoDominance.
On P1. Yes, Maureen, you are correct in a sense that we missed out something in the question when we were typing. It should have said, if 2 black and white cats are crossed. So actually, we missed out something in the question there. If 2 black and white cats are crossed. Now, if we kept it the way it is, then this genotype here should have been capital B, W, W. That would have been the genotype. And in that case, that means all would have been black and white. But we wanted to see whether when we take the heterozygous individuals, what we get. So we did the F2 generation here in this case. Yeah, I'm hoping you're following what I'm saying. Let's go through that slowly. Maureen says that this genotype is incorrect if we look at this question. She is correct in that sense. Problem was we forgot to include here if 2 black and white cats are mated. But if the question stays it's the same, then obviously for black, it would have been capital B, for white, capital W, and our offspring would have all been 100% black and white. We wanted to see when we take black and white individuals, what happens.
All right,
Next
nice
question.
one. Very smart. She's the only one who
Short
picked that
Maureen,
up.
thank you for checking that up. The others are sleeping.
Right, here's a question from Vusi Kevin. What is the difference between the multiple alleles and the dihybrid cross?
Vusi, please understand the first thing. You do not, in the curriculum, need to know about dihybrid crossing. Dihybrid crossing means we're looking at 2 characteristics at the same time. It's not prescribed for the syllabus, so don't stress about that. Multiple alleles, on the other hand, means there are more than 1 allele coding for a particular characteristic. Now, look at dihybrid. Dihybrid, we're looking at 2 characteristics at the same time. Whereas multiple alleles, for 1 characteristic, there may be more than 2 alleles that code for that. For example, For example, blood groups. Blood groups are coded for by A, by B, and by O. But in one individual, there are only two characteristics at the same time. In other words, the person can be homozygous for A, homozygous for B, B, heterozygous for B, BO, heterozygous for A, AO, or homozygous for O, I, two small i's or two small o's there. Okay? At the same time, while you're doing that, you bring another thing into my mind, and that is polygenes. What you should be asking is, what's the difference between multiple alleles and polygenic inheritance? Multiple alleles, where there are more than two alleles coding, but in one individual there'll only be two. Polygenic inheritance, like skin color, for example, there are more than two genes that are coding for that. And the color of your skin is then determined on which one is more of in your body. So there are more than two coding for that particular coded, for that characteristic at the time. Good?
Right, here's another one from Lishiva. Lishiva says, what is the difference between incomplete dominance and codominance?
What's her name?
Codominal, Lishiva. You can
Lishiva.
call her Angel.
Angel.
Yes.
I can't call you Angel Lishiva because you've got to take that coffee mug that's in your hand and knock yourself on the head. That means you haven't been listening to what we've been saying. We started by saying in our example, our first example about complete, incomplete, and co. Let's go through it again slowly. Complete dominance is very easy. One is dominant, one is recessive. When we don't have that dominance, when both are equally dominant, it can either be co or incomplete. For co, the offspring must have both characteristics showing, both of the phenotype. In our example of the cats, black and white both were showing in the cat, so it was codominance. If for some reason, when we blend these two together, the black and the white, and we get another color, or tall and short, and we get medium, or orange and yellow gives you brown, or whatever, whichever way you want to look at it, then it's incomplete dominance. That's it?
Yes, for now.
For now, that's it. Then let's move on. We are busy with question 4. Now we're moving to a little bit higher. Now we did the basics on genetics, how to solve a problem, how to do complete, how to do incomplete. Now we're moving to a pedigree diagram, but this one here just moves straight into it. It has the elements of two things here. One is the pedigree diagram that we have to understand. And two, at the same time, it's talking about sex-linked inheritance. And there were questions on the page about sex-linked inheritance. So those of you who are asking questions on sex-linked inheritance, this is the time to concentrate. A sex-linked inheritance means, for example, one question on the page was, why is hemophilia considered to be a sex-linked disease? A sex-linked inheritance or a sex-linked disease is a characteristic of which the allele is carried on the sex chromosome. That means it's carried on the X or the Y. More prominently on the X, because the Y is too small to carry too many genes. So there's very few that are carried on the gene, on the Y chromosome. More are carried on the X chromosome. So if a gene is carried on the sex chromosome, especially the X, but it could be either, then we consider that gene to be sex-linked. That means it's on the same taxi going to a particular point. Which taxi? The sex chromosome taxi. Don't write that in an exam because you won't get marks for the taxi part. You understand what I'm saying. Okay? So that's what we need to understand there. Okay? Any linkage means they carry it together. And sex-linked means on the sex chromosome. The other question that came on the page is why, when it's sex-linked, more males are affected. And that question comes often in the question papers as well. The males are more affected because if you remember, the males are XY. So they only have one X chromosome. And if this one X chromosome has the defective gene, in case of hemophilia, it's a recessive gene, small h, then it does not have on the Y chromosome a partner, because the Y chromosome does not carry its partner. So the minute this individual has the small h, he's already hemophiliac, as opposed to a female who has two X chromosomes. And if she has one small h, but she still has another X, and if this X has the normal gene, then she will not be hemophiliac herself, but she'll be a carrier of that disease. She herself will not suffer, but she can pass that disease on to others. So you see in the females, that's the story. Let's go straight into the questions now. The pedigree diagram shows the inheritance of hemophilia in a family. X capital H, normal blood, and X small h, lowercase, is a recessive gene causing hemophilia. They're telling you that in the example. Further than that, they also tell you that in the scheme, they tell you when it's clear like this, no hemophilia. And if it's a square, it's a male. Let me not confuse you by drawing a circle over that male. Okay? If it's square, it's a male. And if it is a circle, then it's a female. And if it's clear, generally in all pedigree diagrams, in diseases, the clear one would be an unaffected organism. And the colored one is the affected organism, or the shaded one is the affected organism. In this case here, the shaded one is hemophiliac male. And there's your diagram showing you that. Now, in this pedigree diagram, there's another way of solving it as well. That, remember I told you in that other diagram of the rabbits when we started what to do? Now, before we even look at the recessive, we must look at males and females. Because in this problem, whenever we're doing sex-linked problems, we must use X and Y to solve the problem. So we start by going to the males, and in the area where the males are, we simply write in X, Y. And the females, X, X. So X, X, all the females, and X, Y, all the males. So that's the first thing you do in a pedigree diagram. You write that down, so you do not make a mistake thereafter. Now, they tell you there that the capital H is for normal, and the small h is for hemophilia. And the shading tells you that wherever it's shade, that person has hemophilia. So we go here and we put in the hemophilia there by that guy. So we know for sure that he has hemophilia. Now, what does that tell you about his parents? Does he get this X from the father, or does he get this X from the mother? He can't get the X from the father because the mother can only give X's, not EX, ex-husband, and ex-wife. X's, the letter X, right? So, you with me, AB?
Yes.
Yeah, exactly. Just now the people say I'm saying that the wives are giving only X's. Okay, so the mother can only give you one of two X chromosomes. So she had to give you this X chromosome. The father, for you to be a boy, the father had to give you this Y chromosome here. If the father gave you an X chromosome, then you wouldn't be a boy, you would be a girl, because then you would have two X chromosomes, one from the father and one from the mother. So that means this boy inherited this X chromosome from the mother. So here's the mother here, so this mother must have had the small h. But because she is normal, she had to have the capital H to counteract that H. So she was heterozygous and she's a carrier. You with me so far? Notice how we're working homework first. So that would give us that there. Now, we look at these two parents here. What can we say about these ones? We can't say much here because there's not enough evidence here to tell us whether these are carriers or non-carriers, because this individual did not come from this branch. This individual came from somewhere else. Okay, so this is the wife of number 4, some other family. Good, but nonetheless, we've done our homework. Let's start. They want to know what are the phenotypes of individuals for 1, 5, and 6. Phenotypes. Notice they didn't say genotype. Again here, a typical mistake is when they ask you for phenotype, children write XX and XY and so on. That's not what phenotype means. What is their situation? Number 1, according to the, and again look at phenotype, comes straight from the table or the key that's given to you there. 1 is a normal male. 5 is a normal female. 6, straightforward, is a hemophiliac man. Okay? Then provide the genotypes. Now what? They changed it a bit. Now they want the genotype of individuals 4, 5, and 6. 4, 5, and 6. Now, let's look at 4. 4 was the male. Straightforward. He is XHY. We would write it as follows. X superscript HY. Capital H because he is normal. And number 5, because there's not enough information available, and because they're normal, it's either this one or that one. Remember, we write it superscript or X, capital H, X, small h. In both cases, the person is normal. You can't look at somebody phenotypically and say they're hemophiliac. Their body will not show you that. So there's no sign, so we say they're normal. Okay? And the last one was the easiest one. That one there, we've done already on the diagram. Good. If individual 6 married a woman who is normal, what are the chances of them having a hemophiliac son? Show how you came to your answer by means of a genetic cross. Again, we brought the genetic cross in here. So in individual 6, yes, individual 6 is a hemophiliac male, and he marries a normal woman. Not a carrier, a normal woman. And that's how we would set this question out now. Again, we're not going to waste too much time. Just, again, the P1, the fertilization, the meiosis, the F1, they're all in the diagram. P1 phenotype, hemophiliac, male, father, and mother, normal. Okay? Because you've put father on the top, see there, father, mother, that will tell you that's a male. Or you could say male and female. Then you say genotype, and you give that genotype. Remember the father genotype we've got, and the mother's there. We separate them into gametes. Again, there's your meiosis coming in. And when we have the offspring, this is what we get. There's it there, XH small, Y, XH, XH, and this is what we get. How many of these males are hemophiliac? There's only 2 males there. None of them are hemophiliac. So therefore, our final conclusion is 2 normal daughters and 2 normal sons. Notice here in the phenotype, you must be able to say male or female or daughters or sons because it's a sex-linked problem. You can't just say 2 normal and 2 normal or 2 normal and 2 hemophiliac. You must be able to say the sex, link the sex to the condition to get that story done. Now notice again, using X and Y, the way we set our problem is the same. We only substitute the letters with the letters X and Y because it's a sex-linked inheritance. AB.
Thank you so much. Without any waste of time, it is that time where you just take your cell phone next to you a bit and then go warm that coffee or make yourself a coffee because we're back right after this. Welcome back, Mindsetters. Now about the competition. You either tag yourself to the post or share it on your wall and then give us your lucky number. Aslam's lucky number is 3 for today and mine is 8. So currently our Facebook, our last digit is 6. So I only need 2 more likes to get to 8, and then when the club starts, hopefully it will still be on that. But otherwise, get your friends liking the page. And I have questions for you, Aslam.
Yeah, bring them on.
Right, the first one is from Snentla Ntlatembani. How many alleles control blood type?
The question is very interesting. She says, how many alleles? Notice she didn't ask how many genes, how many alleles control blood groups. There are 3. Which are they? IA, that's for blood group A, IB for blood group B, and small I for blood group O. Oops.
All right, while you're trying to fix that, Mindsetters, make sure that you tag yourself on the post, not on the wall, because I'm seeing a lot of Mindsetters are writing on the wall. So your lucky number should be on the shared post, or either tag yourself on that very same number.
The other question while we're waiting for the board?
The other question is from Matsubane. Wow, welcome, Matsubane. I wonder where have you been today? He says, what are the test crosses and their importance or use?
What is a test cross?
Yes.
And what are they important?
Yes.
What is a test cross, guys? A test cross is when we want to find out the genotype of an individual, we do a test cross. Let's say we have an individual that is coding for red flowers. We want to find out whether that particular plant is homozygous for red color or heterozygous for red color, if it's a dominant one. If it's a recessive characteristic, there's no need doing a test cross, because a recessive characteristic has to be homozygous because of the law of dominance. But when it's dominant, it can either be homozygous dominant or heterozygous. So how do we test this individual then? If we test this individual with a recessive one, and all the offspring come out of the dominant color, then we would know that this individual is homozygous for the condition. However, if in the offspring we find some of the dominant color and some of the recessive color, that means that this individual had to be heterozygous. Why do we say that? Simple. If the individual, say for example, in our example that we are talking about, is homozygous for red, and we are crossing it with a recessive, then obviously all the offspring have to be that genotype, and if it's that genotype, then they are all red. However, if we find some white there, that means in the offspring we're finding some small r. Where did this individual get the two small r's? One obviously came from the parent that we are testing it with. The other had to come from this parent, so this parent had to be heterozygous. So this is a test cross. We want to find out the genotype of a dominant individual.
Right. You were trying to explain the other one, or
Which
are you
one?
done with that one?
We're done with that one.
All right.
Yeah, we're done with that one.
Then we can move on.
Okay, good. Let's move on.
Unless you can take this one. What is the difference between cloning and genetic engineering?
Cloning and genetic engineering, there's a question separately for that, and we will cover it when we get there. Okay, let's look at question 5 while we're at it. Now, a little bit of biotechnology coming in now. We've looked at several examples of genetic crosses to cover us on all those things. Let me just go back, by the way. Oh, that's why the problem was that there was a blank page in between. Okay, we created a blank page. Guys, if this question was given to you with color blindness, how will it change? It would change with the letters that they use. Instead of using H, they're going to use the letter C. Capital C for color blindness and small. Or they could use capital B for normal and small b for color blindness, like the prep paper 2013 Gauteng. They would have given you something like that. So please understand what we are saying there when we're doing that as well. The principle is the same. The way it's inherited is the same. Now, for color blindness and hemophilia in the papers, they don't have to tell you that it's sex-linked, because these two disorders are prescribed in your syllabus. So you need to know that they are sex-linked. If they should give you another problem of another sex-linked disorder that's not prescribed, then the paper must tell you that it's sex-linked. But the way you come to your answer would be the same as this in any one. But be careful to see whether the condition that they're talking about is recessive, like in hemophilia and in color blindness, it's a recessive characteristic, or it is dominant. But the way you work out the problem will still be the same. You'd be using X's and Y's. Good. Now we move on to question 5. The illustration shows the separation of DNA fragments produced from blood samples taken at the scene of a crime, plus those of 3 suspects. There were 3 suspects for this particular crime. So they've taken their DNA blood samples, and they have taken out from those blood samples their DNA samples. And the DNA sample goes through what we call gel electrophoresis, and they form what we call these bands. They form these bands that we are talking about here. And now they give you the bands. They're giving you V for victim. They're giving you the whole story here. V for victim. C for S. CS is sample taken from the crime scene. Suspect 1, suspect 2, suspect 3. They're giving you the pattern of their DNA, or their genetic, or rather, sorry, their DNA fingerprint. This is what a DNA fingerprint looks like. it is not the same as your normal fingerprints. Remember, your genetic fingerprint is from your genes. Your fingerprints are the prints that you leave on anything you touch. Which suspect is most likely to be guilty? And no matter how they ask this question, especially when it's crime, it's easy. You go to it's a simple matching exercise. You look at the sample, and you look at your 3 suspects, and you try and match it. For a start, this one is out. These 2 bands are different, so this one will go out. This one also, the bands are different right across, so this one goes out. That leaves you with that one there. Look at the match here, that one and that one. That one and that one. And so we go down the line. Can you see? All these bands are matching. So our answer would be suspect 3, and the reason will always be the same. Why do you think so? Because suspect 3's DNA is exactly the same as that of the sample. You must compare it to the sample, not to the victim, but to the sample. Why do we need a sample of the victim's DNA? We want to know who did the crime. But now why are we taking the DNA of the victim? This question they like to ask also, and the answer is almost always the same, to make sure that the sample found at the scene does not belong to the victim. We may find blood at the scene. That blood could well be from the victim, his or herself. Now, if we carry on looking for this particular suspect, we're never going to find it because we're going to bury the victim because the victim is dead already. So now it's no sense. We're looking for a needle, not even a needle in a haystack. A needle in a haystack you can still find. Right? But you're looking for something that's impossible to find because you've already buried that person, or you cremated, or whatever you've done to the body. So that's why we need to make sure we have the victim's DNA as well. Question 6. Again from GDE now, this year's prelim. Here's some biological terms. Give the correct biological terms for each of the following descriptions. The term given to the point at which crossing over occurs between 2, that should be 2 TWO, no? 2 chromosomes occurs, and the answer would be chiasma. Notice the plural of that is chiasmata. 1 point, chiasma, more than 1 point, chiasmata. An organism's complete hereditary information encoded either in the DNA or the RNA. His whole story. And that would be the organism's genome. A trait that does not appear in the phenotype when present, does not appear in the phenotype when present in the heterozygous condition. A trait, what type of traits we have? We have dominant traits and recessive ones. They want to know, of these 2, which one does not appear in the phenotype if the organism is heterozygous? Obvious, recessive. Is the one, remember, complete dominance, the recessive one will not show. The cat and the mouse won't show. You can't write mouse because that's not an answer. Banded patterns of DNA, we just spoke about that just now. DNA fingerprint. The disorder caused by a mutation that results in the absence of pigmentation in the human skin and in the hair, and that will be albinism. A sex cell, a gamete. Okay, so you must know your terminology as well. Good. Question 7. This one from February, March 2011, paper 1, question 4.1. Explain, now here's something about genetically modified organisms, and we'll move on with the question. Explain 3 advantages of using genetically modified organisms for food. 6 marks there. Explain 3. Sometimes we can give you more marks for this because you are explaining. So you're going to say a little bit more. We can give you 2 marks for each, so that will give you 12 marks. But in this one, you'll be giving you only 3, 6 marks for each one there. 1 mark for each, sorry. Explain 3 advantages of genetically modified food. The question could also say, explain 3 disadvantages. Oh, they're giving you 6 here, 3, right? So I'm actually reading wrong. Sorry about that. First of all, we reduce the need for use of chemicals or pesticides because resistance of crops to pests has been increased. In other words, when we modify these organisms, we modify them such that they can be resistant to pests. So therefore, we don't have to spray them. So we've reduced that need of that chemical. Secondly, we can select the best genes to produce better resistant crops or stronger offspring to withstand harsh environmental conditions. Remember, the P's there in that sentence are actually ticks that get lost in the formatting. So wherever you see a P in out of place, it's just a tick that should have been there. Using specific genes to increase crop yields. So we can use genes to increase the crop yield, and this, or livestock improvement for food security. We can select genes to increase shelf life of the plant. So the original one was to reduce the use of chemicals. Secondly, to make it more withstanding the harsh environment. The third one is to get a bigger yield, so this will improve the economy. The fourth one is to increase the shelf life. In other words, we want a plant, or well, mainly here in plants, when we put it on the shelf, it mustn't go rotten too quickly, or it mustn't lose its sparkle before the customer gets it. So therefore, we also, and then because of that, there'll be minimal waste, and again, this can be linked to food security. We can select genes that delay or accelerate the ripening of fruit. We want to get this fruit to get ripe quickly so it can be marketed quickly, or we can delay the ripening of the fruit, so therefore we can import or export it or whatever, right? Using specific genes to improve nutritional value. So in other words, we can put something in there that will give the particular food more nourishment. In other words, we could introduce a gene that increases a particular vitamin or a particular type of food that we need. We can also use specific genes to introduce new traits in crops, something new, to suit specific needs of the population. Example, same story as increasing the vitamin A in food. The table below shows the percentage frequency of human blood groups in populations from 2 different cities in South Africa. So they're telling you human blood groups. A, in city 1, it's 25%, and in city 2, it's 45. B, 20, 10. AB, 10, 5. And O, 45, 40. The question says, which blood group has the highest frequency in city 1? You don't have to be a rocket scientist to figure that one out. Look at city 1, look at the highest number, and that answer would obviously then be blood group O. Lowest frequency in city 2, again, no rocket science needed here. And you look at the lowest number there, it should be AB, and that's your answer there. AB. Plot the data in the table as bar graphs on the same system of axes. And that's what we would put it on the bar graph. Notice, same set of axes means both cities must be on one bar graph. And the two comparing the same type of blood must be together, and the others must be apart to make it a bar graph, otherwise it becomes a histogram. Again, look at the rest of it, and I will leave you with this bar graph. Look at that, think about it. When we come back, we will tackle the bar graph in more detail. AB.
Thank you so much. Mindsetters, I just want to urge you to join our online study group. We have a group called Mindset Life Science. So if you are a life science student, I'm sure you are because you're tuning in right now, make sure that you add that group into your groups. Ask some questions. Help other mindsetters also on those groups. Otherwise, after the break, we'll be stopping the clock because currently it's on 1. So if your lucky number, it's still 1 after the break, then you might go through to the draw. But otherwise, we'll tell you what's the lucky number, the last digit after the break. Don't move. Welcome back, Mindsetters. Now we still chat with you on the page and would like to hear from you. But about the competition, remember you have to tag yourself or share the post or the picture that we posted on Facebook, and then you tag yourself on the number, on the lucky number that you have. So unfortunately, we have to stop the clock now. And I'm saying unfortunately because Aslam's number was number 3 and the clock has stopped on number 2. So we stopped at 53,812. So if your number is 2, again, because even on the previous show it was 2, so 2 is doing it for today.
Yeah?
Yeah.
2 is the number.
2 is the number.
Okay, good.
You should have known.
I should have known. I wasn't watching the first one. Good. Guys, coming back to where we were with the graph, notice the heading must be given, and in this case here, both variables, frequency of human blood groups in populations from 2 different cities in RSA. That was it. If they gave you time from May to July, then you must also include that in your heading to get 1 mark. Then you must put a heading for the x-axis. If there's units, you must include that here. This particular bar graph, generally bar graphs on the x-axis won't have units, but if they are at years, for example, then you'll have time and in brackets years, and so too with the y-axis as well. Frequency in population, and notice the unit is given there in percentage. Also notice that you have to show all of that. Notice that your bars are equal width and they are equally apart. The sets are equally apart. This is what you get marks for in the bar graph. So make sure that you have that. They could have asked you to show a cross between different blood groups as well. The only difference in that is when you're using your letters, instead of using normal letters, you will use the letters capital I A for A, capital I B for B, and small I for O, and also capital I, capital B in the case where that's combined. Remember, this is just one allele showing you there. Good? Let's move on. Question 8. And this was something that somebody asked earlier about clones. So here we're asking a question. Clones are a group of genetically identical organisms. Explain 3 advantages and 3 disadvantages of cloning. And you'll notice that your advantages and disadvantages of cloning are similar to that of genetic modification. Cloning, by the way, to answer the question that was asked earlier, is a form of genetic engineering. Genetic engineering is the big umbrella. Under that, we can get GM foods, we can get cloning, etc., etc., etc. Okay, production of insulin using genes from bacteria, from human pancreas, and inserting them in bacteria. That's also a form of biotechnology, etc. in this case here. So you must know that. Good. We move on. What is the 3 advantages and 3 disadvantages of cloning? Very easy. We're going to go through that like very fast, like the Gau train.
No comment.
AB, no comment. Producing individuals with desired traits, the P is for a tick, to eliminate unwanted characters. We find a good organism, we want to make more of that organism so that the ones that have the unwanted characters are eliminated. Better yield to increase the amount of food for a large population, good. Resistant to diseases, to save on the use of pesticides and herbicides, so we save the environment. Organism produced in a shorter time to increase the yield. Saving endangered species, we hear that this particular AB is an endangered species, so we clone some of AB, so we get more of AB, so that means this AB will not move from endangered to threatened to extinct. Okay? Saving and producing body parts, reducing rejection of transplanted plants. AB or Aslam has a problem, kidney problem. We need to make another kidney, so cloning can come into here, where we'll make a clone of Aslam's own kidney or AB's own kidney, so that the body would not reject it. Produce offspring for organisms that are infertile and cannot have their own offspring. Good. Reproduction is not seasonally dependent, and this will then obviously allow us to reproduce at different times of the year. Disadvantages, first of all, and most importantly, people will start with that, objection or religious beliefs to interfering with God's way or with nature, etc. When we start cloning organisms, we actually start eliminating some other varieties, the natural varieties. Therefore, this will reduce the gene pool. So if something happens to that particular cloned organism that affects that cloned organism, there are not going to be other varieties to withstand that, and natural selection is affected in this way. Cloned organism may have developmental, morphological problems and may not survive long. We clone the organism, but something's wrong with this clone, and therefore it may not survive. Costly process, and not all farmers in the country, especially in Africa, will be able to afford it. Cost may generate more experimental waste, causing ethical issues around disposal of waste. may lead to killing of clones to obtain spare body parts. This is now the criminal element coming in and what have you. And again, when we come to human cloning, that has another dilemma on its own. What can a criminal element do if they can clone a human being? And we watch that in all the sci-fi movies that we watch. Okay, the diagram below represents meiotic division of two different cells. Both cell A and B undergo a certain unusual is a key here, unusual phenomenon, and give rise to male game and female gametes respectively. Look at this diagram. The chromosomes are all arranged on one side and nothing on the other side. So in other words, these chromosomes have failed to separate. So these male gametes have extra, and these female gametes have extra also here, and they together forming a zygote. So they've given you that. So you needed to look at that first. The question says identify the phenomenon depicted in cell A and cell B. This is called chromosomal non-disjunction. Disjunction, junction meaning junction meaning joint, non-disjunction, and this in front of junction, breaking the joint, separating it, and non-disjunction would mean failure to separate. That's what this big word means. Okay? In which organisms, plants or animals, does this phenomenon usually occur? It's usually occurring in plants. Most animals don't survive when that doesn't mean that animals don't do it at all, but more often than not, it's in plants. What is this phenomenon generally known as? This phenomenon is known as polyploidy. Why? Because the organism has more than 2 sets of the chromosome. Why? Because we have 2 sets here, 2 sets here, 2n plus 2n gives you 4n, which is tetraploid. What would the chromosome number in the male gamete? Simple. 2n, diploid, or 8. Female gamete, same story, because they underwent the same problem. They'll have 8 chromosomes, or they are diploid, or in that case diploid, yeah, good. And in the zygote, there will be 16. Why? Because 8 plus 8 is 16, and 2n plus 2n is 4n, and diploid plus diploid is tetraploid. What was the chromosome number shown in a diagram above of cell A and B before meiotic division took place? They had 8. They had 8. It's straightforward in the diagram. There. They had 8 chromosomes. State 2 ways in which the phenomenon shown is above is important to plant breeders. In other words, what is the importance of polyploidy to agriculture? And there is it, to production of larger plants, the production of larger flowers, larger fruit, and seedless fruit. Those are the advantages in this particular case. Good. And therefore, that brings us to the end of our questions. We had a few questions there. You want to give me one quickly?
Okay, let's see. Where are genes found?
Where are genes found? There, I'm wearing them. My genes on my legs. Genes are found inside of the cell on the chromosomes. More specifically, genes make up the DNA in our bodies. One last one? Nothing?
Nothing for now.
Guys, I'm hoping that you benefited from the session. Go all the way. Over to you, AB.
Thank you so much, Aslam, and we just want to wish you also a safe journey. Enjoy your holidays. Otherwise, Mindsetters, let's learn more. We'll see you after some other time. Thank you for tuning in. Join our study group online and let's learn more. From us to you, peace.